To me, the whole excitement, what got me into the CRISPR-Cas field to begin with, is my love for non-coding RNAs, right? And how, the RNAs themselves can be the end products of gene expression. There's so many different types of RNAs in the cell that nobody knows anything about. So my whole career has been, all the projects in my long career have all been glued together by my desire to understand the non-coding RNAs and what they do. And so CRISPR just became that for me. It was, and then, an RNA-based adaptive immune system. That was so exciting to me. So that's kind of what got me into it. I mean, in the early days, we discovered the RNA targeting CRISPR-Cas system, the CMR system, right? And people initially thought, how can this immune system function, with RNA? It has to take DNA, so it can't function by cutting RNA. And so that was just a kind of a short-lived controversy, because now we know there are dedicated CRISPR-Cas systems like type 6 systems or Cas13 systems that focus only on RNA, and then others studying these type 3 systems saw exactly what we saw. And so it was just kind of how you conceive of the information in the very beginning with the facts that you have made it hard for some people to grasp this possibility. But this is the weapons used all around the world against all the bacteria in the world. So there's so many surprises yet to go, and you have to be super open-minded about where it goes. But that's the fun. That's why we're all doing it. There's so many surprises around the corner. Yeah. I was 100% non-coding RNAs in eukaryotes, right? And so, if you will, I evolved to bacteria. So I used to work with human cells, with frogs, and there's very interesting biology there, but those systems are so complex, right? And the information it's much harder to obtain. So I was really thrilled, working with bacteria and simple systems, but really powerful systems. I've been in multiple fields, okay? So, and how it compares, it's been a joy, because I was there at the birth of the field, right? Where, in the early days, there was a handful of us around the globe that sort of cared about this problem, right? And we all came from different angles. So there was no competition. It was all, trying to grasp what we were working on in different aspects. And the field has now become sort of more standardized in the sense that now it's bigger, there's more competition, but because of, personally, because of all the friendships we made in the early days and, just really kind of interesting people, I find it to be a very, very great field to be working in. I mean, I really enjoy the community. And now, I've been in fields like telomerase field or cancer field, and now just expand the numbers, and the important questions are going to be looked at by everybody, and it's a race. And so at least in the CRISPR biology field, I feel like there's a lot of room, there's a lot of, but yeah, it's competitive, and there's really, really talented people, and I don't think there's any science on this planet that this exciting where there won't be multiple people trying to go after it. And every year at these annual CRISPR meetings, right, we learn such big things. It's just that just tells me that there's a lot more to go.